Okay, good morning. Let's have a class, and we will talk about the transcription translation today, and we're trying to finish this section. So then go to lactose opera on Thursday. Okay, again, we want to go back to see the central dogma. And we mentioned this is the outline of this exam, three parts of the section. So we had the DNA, do the replication. We already finished talking about that. Then DNA goes to RNA. It is transcription. And then RNA goes to protein. That is translation. And we are trying to finish those two parts today. Okay, first of all, we want to mention what is called transcription. And you need to know the transcription from here is based on the synthesized DNA, and then based on gene complementary policy, we're going to generate RNA, which is mRNA. So what is transcription? It is still code to code. Okay, now what is translation? It is a little bit different. This is code to protein. Actually, it's for amino acids. We know about that. That's called translation. So let's say you're speaking English, I'm speaking Chinese, and then we would like to transfer from English to Chinese. This is called translation. So the same thing here, and we will mention about the details all about this here. Okay. So since we talk about RNA transcription, we need to first of all talk about the RNA first. Why this is not? Okay, so we need to talk about the RNA first. So what's the difference between RNA and the DNA? That will be today's first question. So we write DNA is in here, and RNA is in right here. Okay, what are the differences between them? There are several differences. Number one, what is the structure of the DNA? If we want to draw it briefly, okay. A, H goes H, 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 O, H. We have an H, CH2, and we have a phosphate right here. That is DNA. 2' oxyribose. That's a sugar, 5-carbon sugar, the difference. What does RNA looks like? Basically are the same. Exactly the same. Not much difference you will see. But you will see this 5' carbon, you have oxygen there. And the others looks like very similar or exactly the same, you could say. That's phosphates. Okay, so this is 2' ribose. This is the first major difference is the 5-carbon sugar. DNA, 2' deoxyribose, RNA, 2' ribose. Second, the DNA most of the time is double bonds. And RNA most of the time is single bond. Okay, the third difference. DNA have ATGC as a gene base, adenine, thymine, guanine, cytosine. RNA have AGC, all the same. The difference is the U or difference. So instead of you have a thymine, RNA have uracil. So what are the differences between these two? Let's do these things again. If you remember what's the structure of the thymine, you will understand this very well. Carbonyl group go here, CH3, H, H. This is thymine. Okay, uracil. Let's just write it deeper. also looks exactly the same. So if I have that extra points for you, it is actually just a bonus. To be honest with you, lots of them are exactly the same. What's the difference? The only difference is at carbon number 6. This is a H instead of a methyl group. So some of the people will say the difference between thymine and the uracil is uracil at unimportant place, which is 6 carbon. Instead of you have a methyl group, you have a proton, you have a hydrogen group. Okay, so that's the difference. What else is the difference? DNA, we know it's going through replication, and there is a proofreading process. Or some other fixing system could be to solve the fidelity problem. RNA cannot do that. There is no fixing system, which means error fixing system. This means if you are doing the replication, you already made a mistake. Then you're going to be transferred, go to the RNA transcription. The mRNA certainly will have a problem. Then finally, the amino acids could have a problem, then will become a molecular disease. That's what happened. The reason is the RNA transcription, there is no way to fix it. Last, this is what my feeling, DNA, most likely, it is relatively, we say it's stable. RNA, relatively unstable. Okay, so these are some of the major differences between DNA and RNA. 3, 4, and 5. So that's the first thing we want to mention. Now, what type of the RNA we have? So when we talk about the RNA, there is a major 3 different types of RNA. The first one is mRNA. That is what we call it a message RNA. Since we already talked about the message RNA, what this means, it's carry on the code for amino acids. That's message RNA. The second one is tRNA, and we call it transfer RNA. We will spending a lot of time today talk about that. The transfer RNA is actually carry on anticodon. The last one is all RNA. What this all means? The ribosome RNA. And the ribosome RNA is actually a structure of ribosome. Okay, there is not much difference, not much secret behind that, we can say, because it's a structure of RNA. So that's all the 3 of the RNA. So then we're going to move on to when we talk about what is transcription. Now, transcription is a little bit difference between replication. We will focus on those 3 stages, which is initiation, elongation, and the last one is termination. So very clear, there is a 3 stage, and the same as we talk about the translation. So let's talk about the initiation, what it looks like. Okay, initiation. What is the initiation means? Okay, I have 5 prime. I have 3 prime. I have another 5 prime. This 3 prime. The rounding of the direction. This is DNA. It started from DNA. And the transcription, it will synthesize mRNA. So it also needs to go through a process which is called polymerization. And luckily we can do that because we have an enzyme which is called RNA polymerase. And then we need to talk a little bit about what is RNA polymerase. So we're going to draw it right here. What is RNA polymerase? So RNA polymerase, it is a hollow enzyme, which means there is two components, is a protein components and a non-protein components. And the protein components has 5 units. What are the 5 units? 2 alpha units, beta, beta prime, and omega. And this section we call it a coenzyme. Since this is a protein part, we need a cofactor, which is a sigma factor. Sigma factor is a cofactor. So here we could briefly review, we talk about the enzyme. If this is a hollow enzyme, they're composed by, which is a protein part, is called a coenzyme. A non-protein part, we call it a coenzyme. And the coenzyme was also separately, if this is closely attached, we call it a parasitic group. And if it's loosely attached, and we call it cofactor. And for bacteria, most of the time, if the bacteria survive or grow in a log phase stage, this sigma factor is sigma 70. What means 70? That indicates the molecular weight is 70,000 delta. That's the molecular weight of that protein. So that's sigma 70. Sigma 70 is very important because it helps the RNA polymerase to recognize a very special conservative sequence of the DNA we call the promoter. So RNA polymerase, the sigma 70 cofactor, is important because it helps to recognize this very special sequence of DNA is DNA promoter. So this is the key for the transcription. The double bands of DNA, you must have a special sequence. A very conservative sequence could be recognized by the sigma 70. And then the RNA polymerase can be attached to it and started to do the transcription. So what does that conservative sequence looks like? This conservative sequence, actually there is a lot. But focus on sigma 70, it looks like that. So let's talk about this. We always do +1 is an initiator, or the initial place which happens transcription. On the right side, this side we call it a downstream. On the left side, this side, we call it an upstream. So first of all, about +1 upstream, there is around a 10 base pair, there is a special sequencing called TA, TA, AT. And the complementary, this band is A, T, A, T, T, A. And this is we call -10 sequence. We have a nickname for that called Fry-Bernal box. This is what it looks like. And then from the upstream, there is a little bit 16 to 18 base pair. There is another sequence which is TT, GA, CA. And based on gene complementary policy, this is band is AA, CT, GT. This section we call -35. So the -10 and the -35, this whole section, we give them a name called hormone. This whole section right here, until +1, this is called hormone. And this is a key important place, because this promoter have to be recognized by RNA polymerase. Have to be combined, and they are recognized to rely on sigma 7. Okay. For recognition. So that's what the key at the initiation. Okay, now the elongation. This is the first one. What's the elongation going to happen? Elongation will happen pretty much easily, not difficult. This is a DNA, let's say. Okay, I have a 5' to 3', 3' to 5'. I just write a couple there, okay? ATGC, ATGC, ATGC. This first band, we call it's a coding band. And based on gene complementary policy, the second band is TA, CG, TA, CG, TA, CG, and this is called a template band. Because the mRNA is synthesized based on the template band to do the gene complementary policy. However, when we say it's a coding band, this mRNA sequence is exactly the same as this coding band. The only difference, instead of you have a T, you have a U. So, what's the sequence of the mRNA? 5' goes to 3'. That will be A, U, C, G, C, A, U, G, C, and A, U, G, C. And always remember the elongation 5' to 3'. Okay, therefore it comes out with the function of the RNA polymerase. What's the function of the RNA polymerase? There are 3 functions. Number 1, you have to unwind DNA. This double helix, you have to sneak it. Dig a hole and let's say sneak in, generate mRNA. You have to unwind a little bit. Secondly, you have to read template. This is a template. You have to read it. Number 3, you have to synthesis of mRNA. So this is M1. Okay, that's elongation. Okay, termination. Termination is not very difficult when we talk about the transcription. Why? Terminations, they basically has two types of the rules to do. Number one, when you're going through the transcription, you read, you generate the mRNA. When you go through a rich new area, which means you see those U comes out, it will be terminated. This is what happens. You look at the structure. This is not very stable. The double bonds with these hydrogen bonds on the, sorry, with these protons on the side. Okay, look at this is not good. This compared to the methyl group, this methyl group certainly is much more stable than that structure. So when you go to rich U area, it will stop. Second, this relies on a factor called the rho factor. This rho factor will be meet TUS test is stopped, and very strange, they will have these type of situation comes out. So we call it a stand loop configuration at the end. Then it will be stopped, the termination, and then it will be released. So that's pretty straightforward for the transcription. The key thing here is the initiation part. Now some of you are going to say, what you just told us is different from what we learn in genetics and biochemistry. Because this is for bacteria. And what you learned is eukaryotes. What's the difference? Eukaryotes, the initiator is different. Okay, I don't study for that, but I still remember the initiate is called TA box. Is that right? This is about 30 base pair upstream. Then you go have GC, C, AAT. That is about 50 to 100 BP base pair. That's a sequence. That's a conservative sequence of the promoter in eukaryotes. Second, this eukaryotes is rely on TA factor D, factor B. Is that right? There is a protein for that, for transcription, 2 protein for that. And very important, for the eukaryotes transcription, can they directly go to translation? No, they cannot do it. There is a post-transcription modification. So what happened is, if it's a eukaryotes, you're going to have this sequencing like this, some of the sequence like that, then you have a sequence like this, another sequence like that will be do the alternative arrangement, and this is we call exon, is that right? This is we call exon, and this is we call intron. This is we call intron. So what we have to do is these exon you have to cut. Then these intron you have to connect it, and it will rely on splicing. That is an enzyme can do that. So there is some difference between the eukaryotes and the prokaryotes. So this is what you learned in your biochemistry class of genetics. So we just not going to talk on here, because here we're going to focus on is bacteria. Okay. So, little briefly here talk about the transcription. So we finished talking about transcription. We would go ahead to talk about the translation. here some of the slides which will help you. This is a very important slide, the initiation, the perimeter region you need to know. But I also want to tell you, this is the sigma 70 recognizable perimeter, which, as I said, for the majority of the bacteria function, especially as they grow in the log phase. However, for the others, it's different. So, for example, in the class, when we talk about chemotaxis, motility, flagellum movements, that function rely on sigma 28. And the sequence is different. That is TAA, 15 base pair difference, and then GCC, GATAA. So just want to let you know, this is a model is for sigma 70, is only for standard growth. But when bacteria do the movement and have other capability during their life cycle, the sigma 70 is different. Because every time there is a different mRNA synthesis and expressed with different protein synthesized later on. Their function are different. So for example, if a heat shock response, let's say a bacteria doing the heat cooking process, they have to survive. Their heat shock protein is different. That relies on sigma 32 factor. Now what is that 32 means? That is the molecular weight. That is 32,000 delta. And what is sigma 28 means? That 28 is 28,000 delta. Okay, that's a molecular weight. So just want to let you know, here is only a very standard for normal bacterial growth. And if you don't understand this very well, we have this last picture which is showing you the each steps about the transcription process, how they do it. Okay. But I think you already learned in the other classes, so you should be okay to survive, at least in this section. So we're going to move on to the next one, which is called translation. So we have to talk this out. We talk about translation. I need this whole blackboard to do the to write down all the procedures and explain to you what happened. It's very interesting results. If you really read about these, you will find it fascinating. It's very interesting. There are lots of the chemical structure, and you should be look around that. Because otherwise, you're only going to remember those names. It's pretty boring. Okay, so, move this clearly. We're going to talk about translation. So I already talked about this. The top coding band, coding strand, the template strand.